Good day, viewers. Welcome to JS Learning Academy. This is your presenter, Mr. Mlenga. So I want us to look at this question right here. This is a question that came in Biology Paper 2 for 2011 external candidates. All right. So the question reads, Figure 1.1 shows a plant cell which has been put in a concentrated salt solution. So this is a plant cell right here, which is labeled A, B, C. So this plant cell was being put in a concentrated salty solution, okay? So you must understand when we say it was put in a concentrated salty solution. So we know that even a plant itself, okay, it contains salt, okay? Now here it means it was put in a more concentrated what? Salt solution, a solution with a lot of solutes, but lower water potential. Okay, so that's what it means here. So I'll explain as we keep on answering the questions. So let us look at question A right here. So under question A, identify the parts labeled A and B. So we have the part labeled A, then we have the part labeled what? B right here. Okay, so the part labeled A, this part, this is called the cell membrane, okay? Then this part labeled B, this is called the chloroplast, okay? Now, don't make a mistake of labeling this as a nucleus. No, a nucleus should be dark like this one. So this is a nucleus right here. Now, in case you are wondering, how come this is the cell membrane, okay? So what it means is when a plant cell is being put in a more concentrated salt solution, what will happen is its protoplasm is going to be detached, okay? It's going to be detached from the cells, or it's going to pull away. You can see this cell membrane has been pulled away from the cell like this, okay? So this is what has happened, okay? So this is why this is a cell membrane. Okay, so we have said here A is the cell membrane. All right, then for B, we have said this is the chloroplast. All right, so we have answered our first question right here. Let us now move on to another question. Okay, so we have come to question B. So under question B, we have got two questions. There's question 1 and question 2, Roman numeral 1 and Roman numeral 2. So I'm going to start with Roman numeral 1. What happened to the cell to reach this stage? What happened to the cell? What happened for the cell to reach this stage? So they are asking you now, look at the stage where this cell is. What happened to it? Okay, what happened to it? So what happens is that when this cell is being put in a concentrated salt solution, okay, you find that since there are more solutes there and there is less water potential, so what will happen is the cell, okay, the cell is going to lose what? Water to the outside solution by the process called the osmosis. That is what will happen here. So in simple terms, we are just looking at this cell being pressed in hypertonic solution. So there is a hypertonic solution. Here when you put a plant cell, the cell is going to lose water, okay? And the protoplasm is going to pull away from the cell. But there is another solution which is called the hypotonic solution, where a plant cell is going to gain water and is going to swell and burst. So you have to make sure that you know these solutions. Okay, so right here on B, we have said what has happened is that due to the concentration difference in the cell has lost what? Water to the outside solution by osmosis. Okay, so here we'll say what happened for the cell to reach this stage. So we are saying due to concentration, okay, due to concentration difference due to concentration difference, the cell lost what? Water to the outside. The cell lost water to the outside solution by osmosis. So the moment you answer like this is when you're going to get what? The marks. Okay, you can see here it was carrying 2 marks. So I hope this part is clear. Let us now move on to Roman numeral 2 right here. Okay, so the question is, state the term given to the cell in this condition. State the name given to the cell in this condition. So on Roman numeral 2, they want you to state the name that is given to the cell which is in this condition. Okay, so let us take a look at the cell. So this is the cell. So when you look at the condition of this cell, I said it's protoplasm. Okay, it's protoplasm, meaning the cell membrane, the cytoplasm, the nucleus, it has pulled away from the cell. It has pulled away from this cell. Okay, no wonder you're able to see the cell membrane here. Okay, then this part which has remained, it's the cell. So what name is given to the cell in this condition? So this simply means that the cell, we have cell plasmolysis here. We have cell plasmolysis. So when a cell is being placed in a more concentrated salt solution, or when a cell is placed in a hypertonic solution, okay, it loses water, okay, and its protoplasm pulls away from the cell, okay. So this term we have said, state the term given to the cell in this condition. So this is plasmolysis. Okay, so that is the plasmolysis. I hope we are together. So I've said this is just a situation whereby a cell loses water by osmosis. Okay, meaning as a result, the protoplasm pulls away from the cell. No wonder you are able to see it like that. So let us now move on to Roman numeral 2. How can this condition be reversed? How can this condition be reversed? So this question is just carrying a mark. How can this condition be reversed? Okay, how can this condition be reversed? So here I've said this cell, it has undergone the process called the cell plasmolysis, meaning that it has lost water because it was put in a more concentrated solution, which is hypertonic solution. Okay, so for this condition to be reversed, the cell must gain water. Okay, so for the cell to gain water, it has to be put in a less concentrated solution with less solutes, okay, or with low solute, and that solution is the hypotonic solution. Okay, so how can this condition be reversed? So this condition can be reversed by putting the cell in a hypotonic solution. Okay, so I are saying this condition can be reversed okay, by putting the cell in a hypotonic solution. So when you put the cell in a hypotonic solution, then the cell is going to gain what? To gain water. and the protoplasm is going to be attached now to the cell. Okay, that is what is going to happen. All right, so right here. Okay, so I've said when you put this cell, you put it in a hypotonic solution, because hypotonic solution has low solutes. Okay, it has low solute, or it has less concentration. So what will happen is this protoplasm that was put away from the cell, okay, it will be attached to the cell. It will go back to its norm, because the cell is going to gain what? Gain water. That is what is going to happen. So you have to know these things. Okay, you have to know hypertonic solution, you have to know hypotonic solution. Then there is also isotonic solution, where the concentration in which is outside, and that's all the solution is equal. That is isotonic solution. Okay, so let us now move on. So we have answered this one. We come to our question C right here. Give one reason why the structure labeled C remained in its natural state. Give one reason why the structure labeled C remained in its natural state. So let us first look at the structure that they are talking about. All right. So they are talking about this structure labeled C. So remember this structure labeled C, I said this is what? This is the cell wall. Okay. So this is the cell wall. So give a reason why the cell has remained in its natural state, despite putting this plant cell in a more concentrated solution. Alright, so the reason why this cell has remained in its natural state is because the cell, alright, it's because the cell is made up of what? Cellulose. It's made up of a fiber-like. Okay, so this cellulose, it allows or gives the cell to be able to protect the cell from any damage at the infections. So that's why this structure labeled C has remained in its natural stage, because it is made up of cellulose. Okay, and it is rigid, meaning it is very hard or it's very strong. Okay, so let us answer this. So give a reason why the structure labeled C remained in its natural state. So I have said structure labeled C is made up of cellulose. It's made up of cellulose, and it is rigid or strong. So meaning it was not affected by the concentration. Okay, that's why it remained in its natural state. So we come to our last part right here, which is question D. I believe this is a simple question. Give two differences between a plant cell and an animal cell. Okay, give two differences between a plant cell and an animal cell. So let me talk about these solutions, okay, before we answer this question. I know you know these. So here you must know these three solutions. You must know them. You must know these. You must know there is what we call hypotonic. Then there is what we call hypertonic. Okay. Then there is what we call isotonic. So you must know these three solutions. What happens to either an animal cell or a plant cell when it is pressed in one of these solutions? Okay, very, very important. So for this hypotonic solution, I've said this one has got low concentration because it has low solutes. Okay, and there is more water potential in it. Then this hypertonic solution, this has got what? It has got low water potential and more concentration, meaning there are more solutes in hypertonic solution. Okay, that's why when you place a plant cell in this solution, it's going to lose water through osmosis. Okay, then the protoplasm is going to be pulled away from the cell wall. We call this as the plasmolysis. The cell has lost water, we call that as plasmolysis. Now here where the cell gains water in hypotonic solution, we call that as the cell turgidity, because the cell has gained water, hence it's going to swell and burst. Then you must know when a plant cell is being placed in isotonic solution, because this solution, it has equal concentration. Okay, so this is very, very important. Make sure that you go through, you revise. All right. Okay, so not only a plant cell but also an animal cell. So for an animal cell, we are seeing a situation whereby it's going to undergo cell lysis. Okay, you remember those terms. It's also going to undergo cell crenation. Okay, so please let's make sure that we find time to revise it. Now let us come back to our question, which is right here. Give two differences between a plant cell and an animal cell. So this is very easy. They just want to, and there are a lot of differences between a plant cell and an animal cell. So the first difference you can say is a plant cell has a plant cell has a cell. Wow, an animal cell do not. So a plant cell has a cell, while an animal cell doesn't have a cell. Okay. Another difference we can say, a plant cell has chloroplast. A plant cell has chloroplast, while an animal cell do not. So a plant cell has chloroplast, while an animal cell do not. So this chloroplast, it allows a plant cell to undergo the process called photosynthesis. That is how a plant is able to manufacture its own food through the same process of photosynthesis. So there are a lot of notable differences here. Apart from that, you can say a plant cell is bigger in size, while an animal cell is smaller in size. Okay. Also, you can say a plant cell has a regular shape, while an animal cell has an irregular shape. So there are a lot of differences that you can give. So thank you so much, everyone. We have come to the end of our lesson. Have a blessed day. Bye bye.